Okay, hello to all of you. I would like to discuss this result with you. So this is the picture I have posted on D12. So this is STS page gel electrophoresis, and this is a picture for Rubisco. So this is usually when we do this lab, when we perform this experiment in the lab. So this lab goes for 3 weeks. It's a mini project. In the first lab, we do the ammonium sulfate precipitation. We precipitate out the proteins, and then we do the purification of the proteins by ion exchange chromatography. And the last week, we basically have the samples from we have the samples of all the fractions we have been collecting, and then we take those samples and we mix them with the sample buffer, the loading sample buffer. So all the details are given in the protocol. It contains a DTT, dithiotritol, which is a reducing agent. It breaks the disulfide bonds. It has STS, which denatures the proteins, and also we have the dibromophenol below because the dye is there, so we can track where the proteins are in the gel. And also this is the gel. Okay, so it's a very thin gel, and it is actually inside plastic plates, and we have to run the gel like this is a gel. It's a picture of the gel. It's a very delicate jelly kind of a material, and these are the different wells in the gel. We load the samples here, and then once the gel is run, we use the power supply, we use electric current, and the proteins run in the gel, and the proteins run on the basis of the molecular weight, because you know that proteins are made of amino acids, and the amino acids have different charges on them. Some amino acids are positively charged, some are negatively charged. You know the different kinds of amino acids. But because in this gel itself, the polyacrylamide gel itself has STS. STS is a detergent, and it has a negative charge on it, so it masks the charges on the proteins, and it gives all the proteins negative charge. So in this gel, first of all, the proteins gets denatured because we mix the proteins with sample buffer. It has all the denaturing agents, plus we also heat the sample. So once we take the sample, we mix with the sample buffer, we heat it. So all these things denature the proteins, and you know that proteins, they have a three-dimensional structure. They have a native conformation that's all broken. So the way the proteins are all folded, that folding is all broken, and the protein get converted into polypeptide chains. So on the gel, when the protein runs, it's a denatured protein, and the protein runs on the gel in the form of polypeptide chains. So and all the polypeptide chains run on the gel on the basis of their size, on the basis of the molecular weight, because all the polypeptide chains, they have been given negative charge by STS. So after the gel is run, we stain with the staining dye, which is a Kamasi blue, and then we restain it, and we get this kind of a gel in which we see these bands. Okay, so these bands, so this is like a polypeptide chain band, this is also a polypeptide chain band, these are all polypeptide chain bands which are here. So I'm going to go over each lane, and I'm going to explain you the results. So now, what is in the first lane? This is a molecular weight marker, and this is a picture of the molecular weight marker which is given here. And these molecular weight markers we usually buy from some companies, so they are commercially available, and these are used as reference markers in the gel. So basically, when we buy these markers from the company, they tell us, like, this is a picture given to us by a company. So they have given this picture, and this picture shows that when you will run this marker on the gel, you will get these number of bands, and the size of every band is this, like on the top. So the bands which are on the top, they will be having high molecular weight or high size, and the proteins which are smaller in size, they are going to be running faster through this jelly kind of a material, and they will be moving away from the well. So it's the same principle as your agarose gel electrophoresis. So on the top, you can see the first band, its size is 250 kilodaltons, second is 150, third is 100, fourth is 75, fifth is 50, sixth is 37, and after the seventh, 25, 20, 15, 10, and the unit of the size of protein is kilodaltons. Okay, so this picture tells us when we will run this protein marker on the gel, this band is of 250 kilodaltons. This is 150, this is 100, this is 75, this is 50, this is 37, 25, 20, 15 and 10. Now so why we are running this protein marker? So that we can compare and we can estimate the size of our proteins, what we are looking for. So this is a gel which has been run with the different fractions. I'm going to go over what these fractions are. It's given here. So if we start from here, this is a filtrate. So if you remember from your lab what filtrate is, this is filtrate, this is supernatant 1, this is P1 after dialysis, the P1 pellet, you get it. So the P1 is dissolved in distilled water and it is dialyzed. So this is the sample of P1, the P1 pellet after dialysis, and then this is P1 medium salt. This is the P1. So remember that first day we precipitate the proteins, and second week we pass these proteins over the column, and after we pass these proteins over the column, we elute them using the salt buffers. And we have three different kinds of salt buffers. We have low salt buffer, we have medium salt buffer, and we have high salt buffer. So we collect all the samples, all the fractions, but in this gel everything is not run, because in the second lab we also check the OD with the spectrophotometer, and we do it in the UV range, because some of the amino acids are aromatic. So that's why we take the OD in the UV range. So if the OD is high, then we save those fractions, otherwise we discard them. So that is why every fraction is not run on the gel. So on the basis of if the fractions are showing good OD, only those fractions are run. So this is basically this is P1 medium salt, then this is P1 high salt, then this one is S2 supernatant supernatant 2, then this is P2 medium salt, and then this is, I think, this is empty well. There's a spilling happened a little bit, if the bands we are able to see here. Then this is P2 high, medium or low, whatever you see here, like high salt means this is eluted by using high salt buffer. Medium salt, this is eluted by using medium salt. So this one is P2 high salt, and then this is your marker. This is for our reference. Now we know that what we are trying to do in this lab, we are trying to isolate Rubisco. We are trying to isolate and purify Rubisco. And the size of Rubisco, it is made of 2 polypeptide chains, and the size is 55 kilodaltons and 14 kilodaltons. So if the size is 55 kilodaltons and 14 kilodaltons, so where do we expect the band of Rubisco? We expect this is 50 and this is 75. So we expect the band of Rubisco near this band, near this fifth band. And the second polypeptide of Rubisco is 14 kilodaltons. So we expect the band of second band 14 kilodalton between these last two bands, somewhere here. Okay, so this is 15, this is 14. So if we now, let's go over these and we see that. So this is your filtrate. So you know that what is your filtrate? You basically make the spinach smoothie and you pass through the mirror cloth and you collect the filtrate. And this is a little fraction, a small fraction we have saved of filtrate, and this is what we are running. So you can see here that it is such a big thick smear, and the reason for getting this big thick smear here is because the protein is very concentrated. So that's why not able to see very discrete bands, but you can still see a very thick band of Rubisco here, and the lower band is not very clear. This lane has not run very nicely, and the reason is it's overloaded with protein. So but this band is very clear here. Then as we move to S1, so S1 is when we pass when we do the first ammonium sulfate precipitation with 37% saturation. This is the supernatant, so we spin it down, we have the P1, and this is the supernatant 1. You know what supernatant 1 is. So which means that in the this is filtrate, in the P1 we have concentrated lot of protein. This is the supernatant 1. So you can see how this lane is clear here, and you see one prominent band. Although this lane has not run very nicely, it is like a kind of a tilted, so that is why we do not see the second band at the exact position. This is P1 after dialysis. So this is the pellet. So this is a palette. So it's again very, very heavily overloaded with protein. So that is why you can see it's a thick smear. It's all because of the overloading of proteins. But you can still see a prominent band here. And then after that we move to P1 medium salt. So this is P1 medium salt, which means this is the P1 fraction after we pass through the column and we eluted with the medium salt. So we see one band here, we see another band here. This is P1 high salt. So you see one band here and one band here. So you can see that these preps, these two preps are after we have passed the protein through column. So this is a purified fractions. So these are purified fractions. So that is why you can see how clear these lanes are, and we see some prominent Rubisco bands. And then followed by this is your S2. So in S2 also you can see because a lot of protein has already precipitated out, so we don't see much bands, and you see one band here and one band here. In all these fractions you will see also some additional bands, and the reason for getting additional bands is usually sometimes some proteins do get degraded. Okay, so basically, or it can be like in the fractions which have been passed through the column, we do not expect other proteins in them because that is supposed to be quite pure protein. But in these supernatants, we can expect some other proteins also. We do not know, we cannot say for sure whether it's a mixture of other proteins or it's a degradation of Rubisco itself. It can be anything. But in the fractions which you have collected after passing through the gel, after passing through, not the gel, sorry, through the column, if you see some fractions, it is suspected that they may be some degradation products because we expect there to get purified fractions. In the supernatant, we can have a mixture. Okay, followed by supernatant 2. This is the medium salt. This is the P2 medium salt. You can see here there's one prominent band of Rubisco here. There's another prominent band of Rubisco here. Then this is this is an empty lane. This is just, I think, some protein has spilled from either here or here. That's why we see very faint bands here. And this one is P2. This is P2 high salt. This is P2 medium salt. This is P2 high salt. We do see a very clear band of Rubisco here. 55 kilodaltons, 55 kilodaltons, 14 kilodaltons, 14 kilodaltons. Now we do see some extra bands here. This is one band here, one band here, one band here, one band here. I feel these two fractions should be quite pure because we have passed through the ion exchange column chromatography basically, or we have passed through them. We have passed these proteins through the column. I think they can be some degradation products of the protein, so that's why we are getting some bands, and they are not the they do not have the they do not have the size of 55 or 14. So it's in between 55 and 14. So maybe this product, this peptide, is just a degradation product of this. So that's I mean, we can't say for sure unless we do the Western blotting to identify the proteins with the antibodies. Without that we cannot say, but it is just like a, we can say these can be some possible reasons. So that's all what I would like to explain, but if you have any questions, please let me know. You can send me an email, and for this lab you have to attempt a quiz on D2L.